is Marcel Nelen. I work here as deputy head of the Genome Diagnostics Department, and I'm also heading the core facility of the Radboud University Medical Center, core facility for DNA isolation, sequencing, and the analysis of the data. Of course, you want to have a lab tour, which is impossible today, but I'll show you. So this is our reception. This is where most of the phone calls come in and where most of the problems are solved. The elevator. This is where we have a direct connection with the research lab of the Radboud lab for diagnostics. Samples are entering the department through the third floor, and then when they are collected, they are sent upstairs to the fifth floor where we are located, and then we receive them, and then eventually when the blood samples arrive, they are registered here. They immediately get a barcode so that we don't make any mistakes. This barcoding is more or less the same as you will see in the supermarket stores. We use more or less the same system. So the first machines, you will see a lot of robots in our department, especially in the facility. In total we have like 20 or so of these systems. Every robot more or less specialized. These are the two robots that we have for DNA isolation. The procedure is very simple, straightforward. We receive the blood, as I said, they get a barcode. The barcode is then, of course, entered into the system, and from that moment on the sample can be traced and tracked. We do a very simple DNA isolation. It's more or less a washing street. So what we do, we make sure that the white blood cells are exploding, so that the DNA is released in solution. Then we add magnetic beads, and from that moment on the DNA will be attached to these magnetic beads, and then eventually we can use magnets, which is located here, to fix the magnetic beads where the DNA is attached to the magnets. We can do all kinds of washing steps with ethanol and other solutions, and then eventually the DNA is purified to such an extent that we can use it downstream in our applications. After that, you have to release the DNA from the magnetic beads again, so the connection is broken, and then you make sure that the magnet is on again, and you can take the DNA solution out of the test tube and then transfer it to the test tube that you want to have. This is where the blood samples are stored. Here you see the barcode reader. Here you see all the pipetting tools that are available. And then of course the various robotic systems and pipettes that are needed. And here on the left, this is where the magnet is located. And this is where the DNA isolation actually takes place. Once the DNA is in solution and is stored in our stock tube, we make working dilutions for our downstream applications. We make a dilution of the stock solution, and then eventually they end up in plates like this. Again, we have barcodes on the bottom of the tubes, and we have barcodes on the side of these 96 well plates. With this, we exactly know which tube is where in the system. And we can do a complete sample tracking for all our downstream applications. The barcode might change during the whole procedure, but eventually everything is connected, of course. So overall, it doesn't really matter which barcode we use, as long as we know which barcode is connected to which sample. So this is what we call a capper decapper, and this essentially releases all caps from all test tubes in one go. It's very efficient, very handy, much easier than doing it manually. Then we position the plates here in our robotic system, and then this robot actually makes the dilutions. So we go from a stock solution with very different concentrations of DNA depending on somebody has a cold, or maybe somebody gets some chemotherapy that might have a huge impact on the DNA concentration. So that varies over the whole sample set that we receive, but we want to have a fixed stock solution for our working solution. So we make dilutions to about 15 nanogram per microliter, and then we take what we need when we do our downstream applications like DNA sequencing or PCR or whatever. Depends on what we need. But this system automatically calculates the DNA concentrations and the dilutions needed. And this system here on the left is a 96 well plate absorption reader. There you can actually do all the A260, A280 DNA measurements. And with that we can do all of the calculations needed. Again, here on the side, barcode tracker, pipetting system, and everything that we need to do this operation. Again, robots. So now we're in a next-gen sequencing facility. So this is where we prepare samples for our whole exome sequencing strategy, and probably in the near future we will prepare samples for whole genome sequencing strategies. This is for our in-house workflow, where we prepare samples from, let's say, to get a report out within 14 days, which is quite fast in terms of getting a diagnosis. Essentially it's still the same system as we also showed upstairs. So again, a lot of plates in a carousel. It's now empty because we of course used it yesterday. Oh, there in the back there are a few plates. This carousel is kind of a storage unit for samples that we need during the procedure. Here you see a small door. This door can open, and then you have some kind of a scoop that takes out the plate. And then of course it has to go to a capper, decapper. It has to go through all kinds of pipetting procedures, and again a lot of magnetic bead steps are used to purify and to clean the DNA in between. Again, here a barcode reader. Here on the system you have a PCR system that can do all the PCR steps that are needed. Here you have a system. This is brand new. This is the Covara system. It can shear DNA 96 samples at a time. So if you do an exome strategy, you have to chop up your genome to very small pieces of about 200-300 bases, and then you can actually use it for the rest of the enrichment strategy. Why we have to chop it up is because we use a short read sequencer at the moment. So that makes a big difference in strategies. If the whole procedure has gone through the robotic system, you go here, the plate is sealed off, it's an automatic sealer, and then again it's stored in the fridge. traveling room, and here you see all the equipment that we have used over the past 20 years or so. Kind of an evolution in time, how next generation sequencing evolved. So these two machines here in the back are what we call Sanger sequencing machines. These machines were actually used to sequence the first human genome. We had hundreds of these machines spread out through the entire world. So there were sequencing machines in Cambridge, there were sequencing machines in Houston, there were sequencing machines in Beijing. They were all doing all their sequencing, usually one chromosome at a time. It took more than 10 years to sequence the whole genome. You can argue if it's still finished. But this was the machine that was used. It was actually more expensive to sequence the first human genome than to put a man on the moon. But you can sequence 500 bases to 1,000 bases at a time. It's a 96 capillary machine, so you can do 96 samples at a time, times 1,000 bases, and then roughly 2.5 hours per run. So you can do the math yourself and see how long it will take to sequence the human genome. Of course, you want to sequence the bases multiple times to get a more confident base call. So that's why it took 10 years. This procedure on these machines can now be done on systems like this. This is a system that essentially uses the same type of strategy. You chop up your DNA, you sequence roughly 150 bases, and you can do a whole genome in one day. This was crazy expensive. This is a human genome for roughly 5,000 euros in sequencing costs. This was done in the 1990s to 2000. This is done in 2013. Top of the line that we currently have, this is essentially the same machine as you have over there. The sequencing strategy is the same. The sequencing capacity is crazy more. One system can have 40 of those systems in this machine. So now you can do a genome per hour, which is kind of crazy. If you thought about this 10 years ago, nobody would have dared to say that. But this is what it is. It's crazy expensive. Running a flow cell on this machine costs about 32,000 euros per run, and then you can do roughly 30 genomes per run. So you come up with a price of about 1,000 euros per genome. Essentially the same machine, but 40 times the capacity. The latest development that we have, these are 4 systems in place for long read sequencing. So all the other machines are short read sequencing, so you chop up the DNA in very small pieces. Here you want to have the pieces as long as possible. This is a sequencer that can do a whole genome, but still far more expensive than the short read genomes, but you get more information out of it. You can determine the paternal or maternal alleles in this sequencing, just one sequencing reaction. You cannot do this on a short read sequence because the reads are too short. You don't see the differences between the alleles. So there are advantages over short read sequencing and long read sequencing, and this is our exploration if we want to use long read sequencing in the near future in a clinical setting. So we need to train our technicians, we need to train our bioinformaticians, we need to develop pipelines for this. So it's kind of an innovative way to look at whole genomes, and it's getting the department more and more experienced with long-read sequencing. So that's why we invested in these machines as well. So these two are the main workhorses for exome sequencing. The other four long-read sequences are just for sequencing developments and building up experience for that. If you ask me what's the future of sequencing, I think long-read sequencing will eventually be the preferred sequencing technology. But it needs to step up with capacity and price. So you've seen most of the equipment that we have, not all the equipment, but very much the equipment that we use for our next-gen sequencing strategies and all our storage and DNA isolation machines that you've seen. This is by far the most used equipment, and I think time will tell if these machines will evolve, but I'm sure we will have new systems within the next 2 or 3 years or so.